Start our class. Coming back from spring break, and the last time we saw each other, we had that exam, which I apologize again had took too much time, and I try hard to avoid that, and we can talk about that. One reason I want to avoid it is because that's not what we're being tested on. Right? It's not about, well, how fast can you answer questions? I do want to ask a lot of questions on exams, right? If I just had 10 questions, that'd be frustrating, right? Because you'd miss a couple of them and you'd be like, dang it, I do a lot of stuff. So we cover a lot of territory in this advanced course, and so I want to ask a lot of questions, but it's up to me to guide it so that those questions are, you can see, oh, I can answer that pretty quickly, pretty briefly like that. And so in the next exam, what I would do is there's still going to be 50 questions essentially, but I can double some of them up. So when I ask, for example, draw the PDGF receptor pathway, I can have that be worth 4 points. I don't want to do that too much though, because then it's 4 points is a big chunk of points. If you're like, oh, dang it, I don't remember that one, right? Like that. So it's up to me to try to craft that. So I apologize for that. The next exam is going to be as hard. So looking back at those individual questions, I sent out this answer key video where I talked through it, and I hope that when you look at that video, you're familiar with those questions like that, where you're like, oh, okay, it's a lot of stuff, but we talked about almost all of that in class. Then the next thing I want to talk about is that I think it's really important for everybody to follow up on it, because the final exam is going to cover everything like that. So, and the second exam, the final exam, are going to have a really similar style, with the expectation of what's this, what's that, like that type of stuff. And then I want to describe this. So when I was a kid, I grew up in a farm town in Minnesota, and family farming, and where I went to kindergarten through 12th grade was like not hard at all. Like several students quit school when they turned 16 because they were going to farm, and they were already working on the farm, and they were grown-ups in 10th grade that could work on the farm for the rest of their lives, like that. And in that context, in that setting, there was an opposition to education like that. And so when I started college, I happened to get into a really good college, and when I showed up there, I had never tried to learn anything. No way. I'd never had any homework. It was a fine, town I grew up in, but there's just no way I'd ever had any homework. So I really scrambled, and really scrambled to try to hang in there. And what I ended up seeing, and I know this is flawed, but there's reality to it, is that there were two things going on. One was I was learning stuff, and then the other was playing this kind of game of trying to figure out what was expected in that course from that instructor, right? And so I just figured that out. I just, because I had to really fight like that, because I was not, I was not a good learner. I didn't know how to learn. I never tried to learn like that. So I had to really fight and scramble for that. So I had a really strong attitude where I would study all the time, and I'd study stuff I wanted to study, just learn more, learning more about the course. But I'd also just be like, what is the deal with this course? What's the bottom line? Like that, And I think I've mentioned before that a couple of times in college, classmates would boo me. My classmates would boo, like that, because I would be asking questions in class, trying to manipulate the situation so I could, hang in there like that. So I'd ask questions just to drag my feet like that, just be like, oh, can you go over this again? Like that. So in that setting, I thought that was really kind of a bad attitude, in college like that. But I just had to have that attitude to survive, to just really fight and scramble. And so I don't really like to share it, but I do, a little bit, and I don't want to emphasize it, but in this class, you should be able to see what's going on. And what's going on is it's happening in class, right? Like that, you just have to understand if we talk about something in class, you have to be ready to answer that on an exam. And that's one reason I record it. I'm trying to help you see that, oh yeah, you know, and with this book, and the book is super legitimate. It's used everywhere, you know, this is what universities expect students to learn in that kind of course and stuff like that. But I don't know, all textbooks are organized in a way that's really challenging to sort of navigate and figure out what's the most important thing, what are the take-home messages, how could you, you know, say this thing and stuff like that, right? So we're going to keep trying to read the book, but you know, I pointed on the answer key video, there were just a couple of questions that were from the book. Now, to really understand what's going on in class, you have to have the book and go in the book and dive in there, but not dive in there like page 1, page 2, page 3, but rather look at the book and skim through it. And that's a skill that's hard to do because that seems to me like just kind of slacking off. Like I'm just paging through the book. Look, I'm done with my reading. I can go, you know, to the beach like that. But it's the opposite of that. It's a real active, aggressive learning. And just imagine me again, man, just struggling in college like that. And you know, maybe some of you attended high schools and stuff like that too, where they were just really weak and not preparation. And we have the Learning Center here. We have Riley to support that learning effort. And I didn't know of anything like that in college. I would just, you know, hide out in the library and just be scared like that. And so that's this thing I want to point out without too much emphasis, but it's like you could drive a truck through that, is that you just have to really understand what's going on in class. And then these questions are all just real straightforward. I'm not asking you to add to your learning. Like sometimes in like organic chemistry lab, class, they'll be like, okay, you've learned how to synthesize this. Now how would you synthesize, you know, black pepper, you know, like that. And you're like, oh my gosh, I have to apply my knowledge, right? We're not applying our knowledge. We're just saying, what's this, what's this, what's this? Flashcards really work for that, but there's so many flashcards that they really need to be arrayed out on stuff like that. So you and I can talk about that. So please go through the answer key video, go over your exams, bring questions to class. You know, maybe you have some today, that's great. We can go through stuff, go looping back through it, because we still want to learn that stuff. The second exam is going to build on this knowledge, and you won't have to go back and study for the first exam, for the second exam, but need to understand that, so you really want to understand it, understand the format, and then of course again the final covers everything. So lots of this will be back on the final, and then let me know. Lots of students do really poorly on the first exam in this course, and then they end up doing fine, because I have a lot of respect for somebody that can go, okay, I can figure out what's going on, because that's what I think college is all about. A lot of it's based on my experience, is I had to scramble and figure out what is going on in every class I had. You know, these professors wanted something else, and it was a different kind of learning, a different material. And I just have tons of respect for that. And I think that's why there are different courses in college and different instructors, is to make you a flexible learner. So I can help with that. I'd be happy to meet. And a lot of you are freaked out because you never did as poorly on an exam, right? Like that. But if you can dig in and do better, on the next exams and the term paper, if you have to write that, and in the assignments we have, you'll end up with a decent grade. This is a famously hard course in college, and so we're dealing with that. It has kind of a pleasant title, but it's everybody knows that this is a hard topic, because there's just so much of it, and it's so matter of fact that you just roll through it and go like, this is this and this and this. We don't really have to reflect and pause for discussion or debate, because you're just like, yep, there you go. Okay, so that's that. We're dealing with that. And don't feel left out or left behind or anything like that, because I super sympathize with everything you're experiencing. I've been there, and I'm supportive. So let me know where you're at, and how we can help and try to work. And if you say how can I do better on the second exam, I'll say, well, go back over the first exam and see the style of questions. They're going to be the same style of questions, and then see they're just coming right out of the class. Like that, okay? So that's that for this week. So take time to do that. I should say I have the exams. I'll hand it back to the end of the class, but I already sent last week, I sent out this answer key video and the results, and I described in the answer key video that you got a grade for this exam and how I grade and stuff like that. So I won't talk about that in class, but we can talk about it. If you have any confusions, concerns, just let me know. Riley's coming to class on Thursday. He's going to talk about CAR T cell therapy. The second time he came, he talked about the receptor and signal transduction. This time he's going to talk about what the T cell does, so what the cellular response is. And that's good timing because we're going to talk about that next in our course, not about the T cell, but about cells and how cells respond to signals. And that's going to be in chapter 18, and chapter 18 is about the cell cycle, how cells respond to molecules like PDGF and stimulate the growth and go through the cell cycle, which means grow and divide and grow and divide. And so you should have started reading that, read that this week, and even this weekend start looking into chapter 19, which is about programmed cell death. Oh, a little typo there. Apoptosis is what you may have learned that term out is, and stem cells, like that. Here's the flow. We're in this fourth section, so we did this chapter 17, and then we're doing chapter 18, cell cycle, then programmed cell death and stem cells, and then cancer biology. And this whole section is all built to be a package of the molecular mechanisms of cancer, like that. So it's all tied together. We've already looked at some figures that are in these later chapters because the author just keeps cranking at that molecular level. In chapter 20, we'll talk about CAR T cell therapy. There's a little figure on that because there'll be some stuff about treatment, things like tamoxifen and setrusumab, but it's still just grinding down at that cause and effect molecular mechanism relationship. In the context of where we're at, if you say what's the overall sort of big picture of our course, this is our course as sort of flow. Is we've talked a lot about regulation of gene expression, transcription regulation like that. We've talked a lot about regulation of cell signaling, and then the next thing we're talking about is a lot about the cell cycle. So this chapter 18 that we're dealing with this week is a big important chapter for us, like that. Chapter 17 was a big chapter. We built up to it, and we've actually been foreshadowing into chapter 18 a lot. So just be aware of that. And so regulation of transcription in our course, we had that figure that you've been working on, which is that in gene expression, a big part of the dynamics part of the story, the switch story, is regulating transcription factors like MyoD and estrogen receptor and so forth, and ELK, right? And how are those dynamic? How are those switched on and off? And so we have this long list, and that's what we mean when we say, oh, we're really interested in regulation of transcription, is we do that clay model video of muscle differentiation to understand the framework, and then we just pick on those transcription factors over and over and over. And I'd say that we're basically maybe done with this list, you know, of how these guys switch on and off. Maybe there'll be a couple more, you know, but really we're kind of done with that part of this list. And we really focused on transcription factors. Transcription factors are proteins, and lots of proteins that aren't transcription factors get switched on and off by various mechanisms. So this is also a good introduction to broader pictures of molecular biology and biochemistry. So that's that one. And then regulation of cell signaling. Okay, and in that is chapter 17, the organization of chapter 17, which is organized based on these classes of receptors like that. And I'd say we're pretty much done learning just a couple more receptors that we'll learn about like that. But we'll loop back on both the cell signaling and the transcription regulation. We'll loop back and be like, okay, we're going to add to this, especially in chapter 20, when the authors really are talking about cancer biology and how oncogenes like RAS are going out of control. Well, it's all about that signal transduction pathway like that, okay? And our favorite example of this is still this wound healing. We just love that because it involves signal transduction and then activation of a transcription factor like ELK, and then some kind of cellular response, and in our case, it's this cell cycle switch, right? So that thing that's happening under every scab you'll ever have, that's just really the core of our course, like that. And then we radiate out to many, many different examples, like that, okay? And in the process, we covered them this way, but really in the story, right? The story is the first step. Well, PDGF gets released from platelets, right? And binds to the PDGF receptor in the skin cells. The skin cells respond through signal transduction that activates ELK by phosphorylation. It's doing other things as well, but that's our favorite, right? And then what we'll see is, and we've already talked about this a little bit, is, well, ALK is a transcription factor, and what genes does it bind to? ALK binds to the enhancer of certain ALK response genes or ALK target genes, and they're in the cell cycle regulation story like that. So we already leaned into that a little bit, and that's that package, right? So just under that scab, we have really the core of our whole course. The PDGF signal and the signal transduction pathway, the activation of ALK, the binding of ALK to target genes, activation of those genes, and those genes play a role in the cell cycle. We'll see that ALK activates expression of proteins that are the rate-limiting step in moving forward in the cell cycle. And there's a specific point in the cell cycle where the cells are just waiting. The skin cells are just waiting for that rate-limiting step to be filled in like that. And so ALK gets it done by activating expression of those proteins. They weren't there, now they're there, now the cell moves forward like that. Okay? So this third step is what we're going to talk about, and a lot like that, and it's the cell response, right, like that. So the third step is the target cell, the cell that's receiving the signal, whatever the signal is, right? And it's making a cellular response. So our favorite cellular response is going to be the cell saying, I'm going through the cell cycle. I'm going to grow and divide. That's our favorite thing. That's chapter 18. Cells can do a lot of different things. Some of these signaling molecules are neurotransmitters, like glutamate and so forth. And so those cells do respond by having neurotransmission, by having an action potential. So there's many, many different cellular responses cells can make, and we're picking on this response to growth. Okay? So that's our favorite example, but that's just one of many possible examples the cells that can do to make responses. So in the vast variety of cellular responses, sometimes cells express new genes, sometimes they don't. Sometimes they do both. So there are a variety of different kinds of cell responses. We really like to talk about new gene expression. ALK is going to activate new gene expression, but in some cases they don't. I think in our whole course, we'll just have one example where the cells don't really respond by making new gene expression, and that's this one where we said, epinephrine, adrenaline, activates this fight or flight response, and it causes cells like muscle cells to quickly break down glycogen and produce glucose right in the muscle cells, so that the muscle cell has more energy to run away from the tiger. And you see there's no gene expression happening there. There's lots and lots of examples like that, and lots of examples where there's some new gene expression, but also other things happening in the cell. And our book shows that some, but our book is also emphasizing that transcription response. When Riley talks to us on Thursday, he's going to talk about what the T cells do. And they do express some new genes like that. They make lots of other responses too. One of the responses they're going to make is they're going to grow and divide like that. So it's a good example for us to listen to Riley on Thursday, and what do these T cells do? When they get activated by the signal, what do they do to really fight that cancer, okay? So that's where we're at, that's the flow, and so, we're always down at nuts and bolts levels, but there's a lot of logic and I'll say like storytelling power to this, right? Like you could say, what's happening under a wound, and you could tell half of everything that we talked about in our course. Kind of like that, okay? So that's fitting in. Here's just an image of this cell cycle, and it's called the eukaryotic cell cycle. Eukaryotic cells include all animal cells, all plant cells, and a lot of single-celled eukaryotes, like the yeast that causes bread to rise. And the yeast that causes bread to rise is a famous example because it is in that species that the eukaryotic cell cycle was first worked out genetically. So we're going to learn about regulatory factors like, for example, what genes ALK activates. And it turns out that yeast that causes bread to rise activates orthologs of those same cell cycle regulatory proteins. They're found, orthologs easily found in every eukaryote. And the way to think about that is just that, an ancestral eukaryotic cell worked out how to regulate this cell cycle, and then it's just really critical to regulate that properly. And so any organism that happened to get a mutation in this pathway died, didn't reproduce, and so we see a tight conservation or similarity of cell cycle regulation in every eukaryote, like that. I'll tell you a quick story about that reality. So in graduate school, I had a girlfriend, and we decided to drive across the country to meet our families that lived all across the country. And pretty much the first stop was in western Wyoming, where her dad's parents lived on a dairy farm in a small town that was 100% Mormon folks, so real strong fundamentalist Christians. And so we were staying there, and she and I were both from the East Coast, which was problematic to start with these people. And then we were both graduate students, and we were talking about evolution and all kinds of stuff that they're like, they're not happy about that, right? And so during the day, they would milk cows in the morning and the evening, and during the day they do other chores. And during the day, the mom would make like a loaf of bread, maybe every other day. Grandma would be in there, and Laverne. And I was just being cool, and I grew up in a farm town, so I could help out, help milking cows and stuff. And I walk into the kitchen, and Grandma is kneading the dough for this bread, and her back is to me, and she's kneading the dough, and under her breath, she's chanting over and over again. As she's kneading the bread, she's chanting, "I am not a monkey. I am not a monkey," right? Because we've been talking about this evolution stuff, and she didn't believe in evolution, right? She believed in the biblical story, and she's not a monkey, right? Like that. And it's just stunning to think about. That's Grandma, and her granddaughter is studying yeast in graduate school. That yeast. And I just kind of backed out of the kitchen, I'm not going to go in there and say, "Hey, Laverne, guess what? Your granddaughter studies the freaking yeast that you're kneading in the bread dough, because you are not just a monkey, you're also yeast," right? Like that. And Grandma would like that, right? But, in just two generations, right? Grandma, mom, granddaughter, and like that big a switch in this, understanding, right? We, as part of our trip out west, my girlfriend and I were driving all the way to Seattle to meet a guy named Lee Hartwell in Seattle who was studying this stuff because she was working on it in graduate school and wanted to meet him. He won a Nobel Prize later for studying this. And he was working at a place called the Fred Hutchinson Cancer Research Center studying that yeast. So that's a big part of the story, is that, oh, wow, this is true for all of the cells in our body and every animal on our planet and plants and single-celled eukaryotes like these. And so here's the flow, and this is one level, and we'll see, we'll talk about sort of three levels of talking about the cell cycle. This is one, it's just a visual. It's very easy to draw. And really the cell cycle is linear. It's on a line. The process gets repeated over and over again. The cell grows, divides, So it's repeated, and so people just draw it out as a cycle, a repeated cycle. And so it has these phases that you likely learned about, and names. So we got G1, S, G2, M. And that's the kind of stuff that this Lee Hartwell worked on in yeast and the regulation of that. And what we'll find is that there's a stoplight right there between G1 and S. And that's what PDGF regulates, is that the skin cells are stopped there at a red light, and PDGF simply turns the light green, and they're allowed to go through this cell cycle. Okay? So we'll go through all of this, and today we'll kind of go through some of it in an introductory way of the pieces that are in chapter 18. But we'll always have this kind of visual in mind and thinking about where we're at. Now, this is just a drawing of events that are happening inside of each of our cells. And so the proteins that regulate these steps are just in the cytoplasm and in some cases in the nucleus, just floating around like that. But you can think of this as like a little bit like a clock in every one of your cells. And the clock can be stopped like that, paused, and then goes around like that. Okay? So we'll go to that visual a lot in this. And I'll just skip that and say that when we talk about this, and I think chapter 18 is basically organized into these three parts. And we'll definitely emphasize that. 3 stories in our class. So the first one is the regulation of that red light. And that red light is called start. It has a couple of different names, but start is a great name. And it's like the start of the cell cycle. So it's a good name. There's a red light, it's going to turn green, that's the start. So when we say, what does PDGF do? PDGF regulates start in skin cells. Bottom line, like that. Okay? So we'll spend a lot of time talking about that, and we'll be down at that molecular level of like, what do you mean red light, green light? What's that involve, right? And we'll learn exactly what that is, right? That's just a metaphor or analogy right now, but we'll understand the molecular mechanism. It turns out that the molecular regulation of start is really complicated. And so to help introduce it, we actually go to another point in the cell cycle, and we'll learn about the regulation of entry into mitosis. And so let me just show you that because it's a nice visual. So what we're really interested in is that point right there called start. And it's at the end of G1, and it's before the start of S. And so it's a specific point, and that's that red light, green light point. And there's just all kinds of checks and balances and regulation there. Yeah. So we'll learn about some of those because it's super important and interesting, right? Because that's what goes haywire when somebody has cancer. And to understand that, we'll go way over here to a very different place, and we'll look at the regulation of the entry into mitosis. Because it's a simpler story there. And so we'll get a good feeling for it because there aren't so many steps that you can really see how there's a kind of biochemical, like a little circuit switching things on and off. And so we'll go to that point. And then the third thing we'll cover is that, and the book does this too, is that within mitosis, if you look at mitosis under a microscope, you see stuff happening. And that's something you would have learned about some in intro bio. That's where in mitosis the chromosomes condense and become visible, and they line up. The metaphase plate, then in anaphase they pull apart like that. Microtubules pull them apart. So we'll talk through some of that. And that would be like the cell biology of the cell cycle of seeing, oh, I see what's going on there. Just to show you this, when you look at this, historically, scientists like, for the last 100 years ago, they could look under a microscope and they could see changes in the cell cycle, but only things changing in mitosis. Like that. You can see that under a microscope. You say, look at the chromosomes are appearing. We must be entering mitosis. And then we have prophase, metaphase, anaphase, telophase, right? So there's that little piece. And so that's what you call the cell biology of it. And scientists didn't know this other part of it, which would be more the biochemistry and the molecular biology of it, like that. And important stuff happened during this period. And this figure shows that historic understanding was limited to this. Looked like, I see mitosis, we can study the different phases of mitosis and the chromosomes separating, stuff like that. And then the rest of the cell's time is just called interphase, because it just looks drab. Stuff's happening, the cell's living its life, but under a microscope, just like, I don't know, like that. So to start out with, we have that cell biology, and that is, lots of stuff happening in mitosis we can see, and then this blasé interphase, like that. And then we want to understand what's happening in interphase because there's a series of steps that are like start, DNA replication, and so forth. So those are the 3 pieces that we'll talk through. So today we'll kind of introduce start and kind of link it to the rest of our course that we've been learning, because we'll spend a lot of time talking about that start as part of the target cell response to a signaling factor like that and changing gene expression. And then at some point, maybe a little bit today, we'll say, okay, let's go over to that regulation of mitosis to kind of get a little better feeling for how these are biochemically regulated. And then at some point, we'll just roll through those phases, and we want to get a little bit out of that. Our book spends a lot of time on it. So if you just had our book, you would spend a lot of time studying that. And that's not our emphasis like that. Our emphasis is really ranking them like this. Start is our favorite, like that. And to understand start, we'll look at the regulation of mitosis, good, and then, we'll roll through some of the phases like that. So that's where we're at. And so let's talk and introduce start some, okay? So this is just putting two figures together. And this is actually from Intro Bio textbook, the way it shows it. Just introduce it like that. And so here's the cell. We're inside any one of our cells. And so there's the cell membrane. And there's a signaling molecule like PDGF. And it's going to stimulate receptor, PDF receptor, signal transduction cascade, activation of ALK by phosphorylation. ALK is going to cause the synthesis of proteins that are needed to get through this start point. It gets through the start point and goes through the cell cycle. So this is the visual for what's happening in our course. Is we're taking chapter 17 and we're just going to link it to chapter 18 based on that visual. And this is all stuff that's just floating around in the cell like that, but that's the link. It's very physical and very understandable. There's just so many steps that we really have separated them, right? We really said, we got to talk about transcription, start our whole course and really understand transcription. There's a lot to understand there. And then we can say, one of those transcription vectors is ALK, and ALK regulates the cell cycle. We want to get to that. So how does ALK get regulated? We're like, well, a lot of times it's an external signal that causes ALK to be phosphorylated and activated. So what's up with that? And then what does that mean? What does it mean? Oh, it's going to stimulate the cell cycle. So that's the flow, and it's targeting that point that's called start, like that. Okay? So this woman who has skin cancer has a problem with that pathway, right? She's making her own PDGF, and so she is just constant.
Is start different than the restriction point?
No.
No. Okay.
Yeah, and so the book starts out describing the cell cycle, right? And the book is often ranging over a lot of territory. And I'll just say that that start is, let's just write that down. So start is synonymous with the restriction point. And why isn't this getting going here? My little pen is not doing anything yet. What is that? Sorry. Checked it. There we go. So let's just go to this and say that if you learn start, great. So there's start. And start is at the end of G1, and it's before the start of S. And S stands for S, it stands for synthesis, because it's when DNA synthesis occurs. That's when the chromosomes get replicated, get duplicated. And we need to do that because we're going to divide the cell in two. So we need to replicate the chromosomes and give each of the, what are called daughter cells, the same set of chromosomes. So there's start, and start is the best name for it. There's another name that is a really functional name, that's a really nice name that I like to say, and it's called the G1 to S transition. Because biochemically, when you say we're going through start, what you're really saying is we're going to start replicating DNA. So if you look at what happens when the light turns green, the cell starts to synthesize a lot of the nucleotides, A, G, C, T, because it needs those to replicate the DNA. So the cell is, when you say, what is a green light, what does that mean? It just means the cell is activating the machinery to copy its DNA. And there's no turning back. It's just going to roll into that. like that. So the G1 to S transition is a great term. The book wants to use this term, and some of this is like historic, like that Lee Hartwell, he's still alive. We could go party with Lee Hartwell right now. And that was originally called the restriction point. And in our book, I think our authors sort of lost track of what the real reason is why it's called the restriction point. But it's just satisfying just to hear it once, and not a big deal. I don't need you to learn all those terms. Start is great. But just to say it, it's really interesting to hear. So these guys were studying yeast, and it's the yeast that causes bread to rise, right? And so you can just take some yeast, add some sugar, stir it together in warm water, it just starts to grow really fast, right? So it's like responding like the woman with skin cancer, it's just, let's go, right? And so it starts to grow and divide, right? And so they just looked for mutations in genes that wouldn't grow properly in response to warm water and sugar, like that, okay? And they'd say, well, those probably are involved in this cell cycle. So we start studying those genes. That's what they were doing. They were geneticists, this Lee Hartwell's geneticist all the way, like that. Look at mutations, look at that mutant phenotype. Oh, they're not growing. What's the problem? Like that, okay? So if you think about that, those mutations are actually a little bit difficult to work with because the yeast is going to be dead, right? And you really want to be able to propagate that, grow that, maintain it. So how could you study that? And most of the time what they did is they used mutations that are called temperature-sensitive mutations. And it's just, in this kind of, nuts and bolts course, it's good to hear about a temperature-sensitive mutation. So what they would do is the yeast can grow okay at some different temperatures, right? It's like a cold-blooded organism type of thing, and you could raise it at some different temperatures. And so they would pick a temperature like, say, 33 degrees Celsius, and they would be growing the yeast there, and then they would raise it to, say, 38 degrees Celsius, and the yeast could still grow. But what they would do is they would look for mutations that wouldn't grow at 38, but would still grow at 33, like that. And the way they did that is they could grow them at 33 and grow them forever, maintain them, study them, and all they had to do was just shift the temperature up to see the problem. And what's going on there is these mutations, temperature-sensitive mutations, are in coding genes in the coding region. And they make a protein with a mutation on one or more of the amino acids, and the protein is just a little bit floppy. So that at a low temperature, the protein is stable and it's in its functional shape. But if you just raise the temperature a little bit, the protein is basically really sensitive to denaturation, and it just unfolds a little bit, and it doesn't do its job, right? And so this lower temperature, it's just a laboratory technique, right, to be able to get through that work. And they call the lower temperature the permissive temperature, and the upper temperature the restrictive temperature. And that's actually the original reason why they called it the restriction point, is just based on this somewhat convoluted genetics, where they say we're going to switch to the restrictive temperature, and then we see them stop, and they stop at the restriction point, like that. That's the original use of it. People use, kind of apply some different expectations to it now, like they could call the restricted point because there's no growth factor, there's no nutrients like that, and it's restricted, can't grow yet. But that's the original reason, and we don't need to understand it that in great detail. I just want to do that to underscore that the original work was done in the yeast that causes bread to rise, and that's just nuts. The person won a Nobel Prize in medicine, and you're just like, whoa, right? It's got nothing to do with anything except, oh man, they found these same, orthologs in us. So it's one of the most famous examples of how These are orthologs, and the understanding is that there's an ortholog for each of these regulatory genes in humans and yeast, and it's because we had a last common ancestor that already had that gene, it was already playing that role, and it just had to keep playing that role. Anytime it got mutated, those organisms were dead, like that. Yeah, Hannah. The restriction point is synonymous with start. It's the same thing. And the G1 to S transition, it occurs in G1 at the end of G1. Yep, Ellie?
Are you saying like for the mutations, like those are just more sensitive versus like the regular initial genes with temperature?
Yes, yeah, that the wild type or the starting genes cells you could grow at either temperature. Yeah, that's a good question. Yeah, Godwin?
So at higher temperature, were there any mutation that grew better at lower temperature?
No, you can set it up in different ways, but they typically would set it up where the higher temperature, and you think about it, the higher temperature, maybe it's just too much and the protein starts to get unfolded. And so they would set it up where that would be the restrictive temperature. They could not grow there. The mutant could not grow there, but it could grow at a lower temperature. And that's a little bit obscure, but it's molecular genetics, and like people like me just love. I drove across the entire United States with my girlfriend to go talk to this guy, right? Like that, and we're like, that's gonna be cool, so cool. We're gonna hang out there, and oh yeah, that's gonna be great. Yeah. And just diving that a little bit, just to help us focus on that start. And if you call it start, great, like that. Okay. And so that's that, and that's the problem with that woman. The woman just has that pathway just growing too much. And typically, when a person has cancer and it's growing out of control, it's this pathway, somehow a regulation in this pathway. RAS mutation, ERB, blah, 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 and we're just getting the light turning green too much. Okay? The start is happening too much, okay? So just to show you, to fit this in, we'll talk about start a bunch, to just show you these other parts, just to introduce them. So these red marks, again, a figure from Intro Bio, these red marks are really the three most famous places where the cell cycle is regulated. And so we want to learn a little bit about each, mostly about start, a little bit about this one, which is the what's called the G2 to M checkpoint, and just a tiny one about this one, and it's called the M phase checkpoint. And checkpoints are where the cell stops and makes sure everything's okay. And for Intro Bio, I put these images of a washing machine on here, because the washing machine, when you use it, goes through a cycle, and then it's done. And then you do another load, and it goes through these steps, right? And the steps are, on a washing machine, you push start. So I think I have a little figure of that somewhere near here. Yeah, there it is, right? So think of this as being a washing machine where PDGF is pushing start. And then you're just going to go through the whole thing and stop here again, like that. So that's start, and that's our big focus. Then, when you have a washing machine, I know this is a little bit silly, but it's a good analogy. If you leave the lid up, it goes a ways, and then it stops, because there's a problem. And so, normally you push the button, the washing machine fills up with water, it agitates the clothes like that, and then it's going to spin. And somehow the manufacturers decided that's kind of dangerous. Like maybe a cat or a baby could fall in there if the lid is open. So it's okay for it to fill with water and agitate the soapy clothes, but then we better have this stop if the lid is open, and just wait. And that's what this G2 to M checkpoint is. A lot of people call it a DNA damage checkpoint, because that's why the cell stops. Is if there's DNA damage, the cell waits until it's fixed. We're going to call it the G2 to M transition because that's really what's happening. The cell is going to go into mitosis, but it's waiting to make sure everything's okay. And that's very much like the lid up on a washing machine, right? It's just going to sit there until you come and close it. There's a problem that needs to be fixed, and it's just going to wait till it's fixed. In our case, the problem is going to be DNA damage. If there's DNA damage, it's like, whoop, I'm waiting, I'm waiting, I'm waiting. DNA damage is fixed, I can go forward. So think of it that way. And then there's another thing that can happen to a washing machine that's really weird, and that is if you have an uneven load and it does a spin, so it's just shaking the washing machine and it's like moving it around on the floor, sometimes that gets so extreme that the washing machine turns itself off. And that's a good analogy too, because I don't know if you've ever even experienced that, but it happens, and it's really, really rowdy when it's off balance. It's like that. If it's really bad, washing machines will stop and wait till you figure out and balance the load, right? So that's what's happening here, and that's this M phase checkpoint, and we're just going to talk about it maybe only now. Our book talks about it a little bit, but maybe just once. And what this is the cell checks to make sure the chromosomes are getting lined up properly and separated properly. And if they're not, it waits to try to fix that problem. And that's called the M-phase checkpoint. It might be the only time we talk about it. The reason it comes up here is because, we'll mention it again. The book talks about it in chapter 20. We'll mention it again. The reason it comes up here is cancer cells often have problems with that checkpoint. They often have problems where they've lost the control and they don't really care if the chromosomes get separated properly. Cancer cells, like an advanced stage cancer cell, really kind of maybe typically have the wrong number of chromosomes. And scientists have known that for years and years. Too many, too few. And that just arises as just part of just the problems with cancer. And when you have the wrong number of chromosomes, the cell gets even wackier. So just a little bit about M-phase checkpoint. And it's the check-in to see the chromosomes are getting properly separated. Half should go to here, half should go to there. Are you doing that? Like that. So really 3 points like that. Okay, most of these start. We'll go over there and deal with that G2 checkpoint, that G2 to M transition. Then there's phases of mitosis. Ooh, I lost it. There it is. The book has this kind of thing, right? And we'll get back to this, but we'll just learn these names of the phases of mitosis and then one of the things that occurs in each one of them. You don't need to scramble to do that now, but just to say that's the flow of that. It's a cell biology. It's a visual thing, and we'll learn about steps. And what we'll really try to do is learn about a couple of things that happen in each of these, just a couple. There's a long list of things that happen, but just the highlights to walk through that. Because again, not the most important thing for us. So when you look at chapter 18, you can read through that part of the chapters, but breeze along looking for highlights like that. Yep. And so that's where we're at, okay? So we're going to start a little bit to help link it to what we've been learning. And so we'll get more of a feeling for it. Then we'll leave it behind for a short time to talk about that G2 to M transition. And then that'll help us understand when we go back to G1 to S transition start and learn it a little bit more, okay? So like I showed, the woman has a problem with start. There are no mutations in the start mechanism. There are no mutations in the signal transduction pathway. Her problem is she's just making PDGF. So she's just standing there pushing that washing machine button. She doesn't care if the clothes are in it or not. She's just constantly doing that, right? Effectively thinking, oh, I need to heal, I need to heal, I need to heal. But doesn't need to, and just grow and grow and grow. And I mentioned before, if those cancer cells spread, they spread with that problem. She's in real big trouble, because they don't need any other cell to tell them grow. They're just telling themselves in this autocrine fashion, grow, 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 push that button, go, go, go. And what's happening inside the cell is the light's just turning green really frequently, like that. It's just like, okay, good, go, go, yep.
Can we review what the problem is with that? Like, where?
Yeah, sure, yep. Yeah, and we should, right? We should. because we're doing this spiraling learning, right? Moving back. Okay, so what is her problem? So it starts out with genomic equivalence, where every cell has all the genes. So she actually has the PDF gene in her skin cells, we all do, but is not making it. Should not make it there because it stimulates the skin cells to grow and divide. So we need to have some kind of control over it. So don't make PDGF in the skin, even though you have the gene. Don't make it there. We'll make it in the bone marrow. And the PDGF will get packaged into platelets in the bone marrow. And platelets are just little packets of a part of a cell that contain clotting factors, PDGF, a couple other things. And they're just floating around in our blood all the time, like a little suitcase floating around. And where there's a wound, the platelets are stimulated to release, and they most famously release clotting factors, cause the cells to blood coagulate and form a scab, and then also release this local paracrine signaling factor PDGF. Doesn't spread very far, and it just is in that space. So underneath the scab, there's some PDGF that's been released from platelets. And so it's extracellular, it's outside of the cells, and all of our skin cells all the time have the PDGF receptor. So in normal healing, the PDGF binds to the PDGF receptor and activates a signal transduction pathway. The PDGF receptor is a receptor tyrosine kinase pathway. We learned that those receptor tyrosine kinases can activate a couple of different pathways. Our favorite is that RAS pathway. So under a scab, the platelets have released PDGF. The PDGF is bound to the PDGF receptor on the skin cells and activated RAS. RAS is active, and it activates RAF, MAP, MEC, ERK, ELK, and ELK is phosphorylated and activating gene expression like that. So all those things are active under a scab like that. Okay? In her, what happened was this DNA damage where DNA, 2 chromosomes kind of got torn at the same time, and the cell, in a natural way, tried to glue them back together. And the analogy there is you drop your bio textbook, it breaks in half, you just glue it back together. It's fixed. But unfortunately, and it must be very rare, you drop both your bio text and your math text. You tear 2 chromosomes and you try to repair them, and you glue your math textbook onto your bio textbook. And now you have this really weird thing. And when we zoom in on the repair that she makes, she's created what's called a chromosomal translocation, where 2 chromosomes got broken, she grew them back together, and what she did is she glued the regulatory region for the skin collagen gene onto the transcription unit for the PDGF gene. PDGF gene should not be expressed, but now it's gotten connected with the regulatory region of a gene that is expressed in the skin, so she just starts making it like that, and just making it all the time. Because collagen is this connective tissue protein, and you gotta make a bunch of it all the time in skin. And so she's just doing that. That's her whole problem. Yep.
I just have a quick question about that.
Yeah.
As you said, her cancer is autocrine, but the PDGF is paracrine, typically in that signal pathway. So can you have both?
Yeah. So in that specific case, it is true that PDGF is a paracrine signaling factor, and in her case, the signal is just self-stimulating. So that's called autocrine. And so we showed, and I mentioned when I was in intro bio in college, I remember thinking, well, that's a weird figure, because it's always shown this way, and it's shown this way in our textbook, where there's just a little cartoon, and it says this is an endocrine response and an endocrine factor, and endocrine factors are hormones. So hormones go throughout the body. And then it says, and that's different than a paracrine factor. Paracrine factors are local factors. They just go a little ways. And that's what we need for PDGF. We just want it to be, spread just under that scab and not spread anymore. Paracrine factors are unstable. That's what's going on there. They just don't spread far before they're destroyed. So there are a lot of paracrine factors, especially in embryogenesis and development, like that. Together, these would compose all signaling molecules. All signaling factors would either be an endocrine factor or a paracrine factor. And then the odd thing is just down here, the figure, they always just say, oh, and look at this. This is an autocrine response. And it's odd. It's an odd thing because it just shows a cell making a signaling molecule that the cell responds to. It's like, why are you doing that? You know, it's like, it just seems you know, like, couldn't you have just kept it all internal and just been like, I gotta get this done, like that, you know, note to self, like that, you know. But it's like, no, I'm gonna make this thing, right, and it's gonna be a reminder, like that. So think about it. It's a different thing. It really shouldn't be on the same figure, because you could have an autocrine response, and if you did, it would also always also either be a paracrine response, and that's her case, or an endocrine response. And in the lab part of our course, that's what I think is happening in my insect that I study, is an autocrine response in this prothoracic gland, which is a hormone-producing gland. Yep. Okay? And so here's a figure from chapter 18, and look at it, because it's that thing that I show my intro bio course, but it doesn't have that visual. So it's this.
How are you guys doing?
It's showing
Are you still working on your questions?
that, but it's not drawing it out, it's just writing it, right? So it's showing that, and it's saying, you've got growth factors like PDGF kicking that RAS, RAF, MEC, ERK, ELK pathway, leading to the synthesis of, and you don't need to remember D-type cyclins, just the word cyclin. Okay? And so what's that saying? Is that ELK gets phosphorylated and binds to target genes and activates expression of those target genes. And what is one? Cyclin. And cyclins activate a protein that controls starch. Yep.
Can you say that again? So ELK is activating cyclin?
Yep. Yeah, and how's it doing that? So, see, this just puts everything together, okay? Because you know, we have our thing where we could draw PDGF receptor, RAS, MEC, RAS, RAF, MEC, ERK, and then ALK. And ALK gets phosphorylated. There's ALK. It's a transcription factor. So ALK is going to bind and regulate transcription of some target genes, some responsive genes to ALK. And what is one of them? It's called cyclin. And cyclins are proteins, so it's a coding gene, and it's going to get expressed, and it's going to code for a protein like that. And so there's a cyclin protein. And there are a family of those genes, and they're given different letters. So D, E, A, like that. Don't worry about the different letters. Just cyclin, okay? And then, so we're going to have cyclin. And then, over here, let's say, is, in this box right here, is a protein that you'll see is going to be our favorite protein in this chapter. And it's called CDK. And there's a family of CDK genes in us, and those each have numbers. So CDK1, CDK, so forth, 4, 6, 5, like that. Don't have to worry about those. CDK stands for cyclin-dependent kinase. So we know a little bit about what that is. CDKs are enzymes. Kinases are enzymes, and they phosphorylate targets, proteins, molecules, add a phosphate, right? Like that. So that's a CDK. That's what it looks like. And what we're saying is that CDK, to be active, requires the cyclin to bind to it, because it's a cyclin-dependent kinase. So one thing that has to happen for this to be active is that the cyclin needs to bind to it. And in the skin cells, the cyclin isn't being made. And ELK activates expression of cyclin. We get the protein, it binds to the CDK is activated, and that is literally then when the stoplight turns green. That is literally what we mean when we say that at start, the red light turns green. We mean just that this CDK is activated. And it's an enzyme, so it has lots of targets to activate. And some of the targets include the enzymes that are going to synthesize the nucleotides, A, G, C, T. Because I said that's something you got to do is you got to make a bunch of those. So this CDK gets activated, and it just increases the readiness to go into S phase and replicate the DNA. Okay.
Wait, sorry, was the CDK?
Yes, yeah, absolutely.
The green light? Is that what I
Yes,
heard?
that's the green light. There's a stoplight, right? And the cells are just waiting at a red light, and then it turns green. And what does it mean that the light turns green? CDK is activated. And we're going to learn that there are, at start, multiple ways to regulate CDK. One of them is you got to have cyclins. So that's one way it's regulated. In that sense, under that scab and in our skin cells, the rate limiting step in their growth and replication is they don't have enough cyclin. If we made cyclin, they would go. So under every scab we ever have, the skin cells are making cyclins, and the red light is turning green pretty frequently like that. Yep. Yeah. Yeah. Right? Yeah.
Is it kind of like it's like becoming more green as more cyclin is being made, or?
That's a good question. The answer is no. It's really like a threshold story where it's really like a red light, green light, where the cyclins start to build up. And we'll see that over at that other place we look at. We'll see that the cyclins are slowly building up, and they reach a threshold, and they're plenty, like that. And again, we'll learn there's a couple other ways, there's other ways to regulate it, but this is just the first one, and it's a good one, and it's a really important one, right? And so that's what's being shown here, just in this kind of simple drawing. And it would be great to really draw that out more fully, like visually, to appreciate that that's what's going on there. Synthesis of these cyclins. Why? Well, because it's a gene that needs to be expressed. And how's it going to get expressed? The gene's not changing. It's this ELK that gets phosphorylated, binds the enhancer, and says, make some of this, like that. Yep?
So you mentioned that CDK phosphorylates something. What is it, phosphorylate?
Yeah, so CDK phosphorylates lots of targets that, when you say, what does it mean the light turns green? I say, it just means the CDK is active. And then you say, okay, but what does that mean? And it phosphorylates a lot of targets, and they're all involved in going into that S phase. So they're all involved in DNA replication and DNA synthesis. So one thing that happens is we need to synthesize the AGCT nucleotide building blocks, We need to activate DNA polymerase. We need to show the DNA polymerase where to start. Where do we get this DNA copy like that? It's all about that. And that's why when the light turns green, you're just rolling into this S phase. Yep.
Is the cyclins have different types? Is that the D type?
Yeah, and we don't need to dwell on that. There's some different cyclins, and the story is complicated, right? Because we're taking it and we're just building the most, minimal, direct example. But it's, there are more checks and balances. We'll learn a little bit about those, and more complexity because, this is all the different normal regulation of the cell cycle that happens, throughout our lives like that. So there'd be different examples in different contexts. And so what we're learning isn't wrong. It's just like the minimal sort of entry image of what's happening. And it is like, remember how I said like it's like dominoes falling, and now we just got this set of dominoes, right? PDGF binds a PDGF receptor, RAS, RAF, MEC, ERK, ELK binds to the cyclin gene, activates cyclin binds to CDK is activated, phosphorylates a bunch of targets that are directly involved in DNA replication. You know, there's no point where you stop and say, then there's some magic. You know, there's some sparks. None of that, right? Which is weird, you know, to think about that, but that never happens in a car either, right? There's never a place in a car where you end up, and then there's some magic, right? Because it's just like chunk, 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 like that. And there's more to it like that, but that's it. So we should have a quick welcome back from spring break chat, and then I'll get these exam handed back to you so you have your copy of that too. So let's get together and do a little concept chat about 3 things you thought were important today. And here's the sign-up sheet. I didn't get that sent out yet, so we're handing around the sign-up sheet. And remember, you should try to sit down with 2 people. And maybe there's somebody that you didn't even talk to yet in this course, so go talk to them and say howdy. And if there's 3 people, then you can split back up. But there's one group of 3 people, and that's fine.
This is real things there. So that's definitely cool. Yeah, it is like 2 different ideas in a way. I agree. I think going that restriction program with G1S transition, those were awesome. Those I thought it was interesting that when he brought up the like permissive and restricted temperatures on yeast, how those temperatures why having some of how like potency and nature are stable or stay stable. The yeast itself. So that was interesting how there was like a temperature component to It's pretty simple.
Yeah, I thought it was good with them when he was talking about the inject form. He was saying that like self-checks.
So we'll just do this another minute. Okay, Tiba, what is one thing that you 2 talked about that you thought was important in class today?
Yeah, we talked about the regulation of start and how like PDGF receptor, activates ALK and ALK then bind and regulate transcription and cyclins, and then cyclin binds and activate the protein CDK that controls starts.
Yep. Yeah, and you see that pathway is just like click, 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 like dominoes like that. But we had to learn about each one of those. Like, what does it mean when you say ELK gets activated and it's a transcription factor that binds to the enhancer of cyclin? What the heck does that mean even, right? So we had to know that so we could just quickly go, oh, I get that. You don't have the cyclin protein. You have the gene. You need to activate the gene. It's a coding gene, code for that protein. So you're going to get its RNA synthesized, right? RNA polymerase binding, thanks, somehow the ELK is helping RNA polymerase get that job done. making the RNAs, the mRNAs, binding the ribosome's translating the cyclin protein, and it binds to the CDK, cyclin-dependent kinase. Need to have the cyclin to get active, binds active, and the CDK starts phosphorylating target proteins. Let me just show you this. This is just the Wikipedia page for a restriction point. Again, it's start, G1 to S transition. And it just says in here, it says the defining, I just choked up. This is the coolest stuff. I'm sorry we have to take exams on this. You know, it would be so sweet to just talk about it and like get tattoos about this crap, because it is such good stuff, right? It says the defining, right? The defining biochemical feature of the restriction point start is the activation of G1 to S and S phase cyclin CDK complexes, which in turn phosphorylate proteins that initiate DNA replication, centrosome duplication, and other early cell cycle events. It is one of the three main cell cycle checkpoints, the other two being the D2 to M transition, the DNA damage checkpoint we're going to learn a little bit about, and then this spindle checkpoint. The spindle is the mitotic spindle when we're pulling the chromosomes apart, like that. So, right, it's just, we're right in on it. Okay, great. So, Ellie, what is one thing? Oh, Joe.
I just have a question with start restriction point. Why is our book breaking up into two different things? Because they had start with yeast, and they were saying that it was like dependent for yeast to go from the G1 and commit to S, that it was dependent on nutrient. And then when they were talking about the restriction point, they were using animal cells and saying that it was dependent on the presence of growth factors, whether they went from G committing to S or if they went from G1 to G0.
Yeah, so I'm going to say that the reason is that they've kind of, you know, so these are people that have been working on this for 50 years like that, and they're thinking about each other as they write it. And so I'm going to say that our author still wants to give a nod to the yeast people like that, and is kind of using this, like the term restriction point has sort of morphed. Like I said, originally it was about this restrictive temperature of temperature-sensitive mutations. And then people used it to say it's restricted like they don't have enough nutrients, kind of thing like that. And so it's sort of slightly morphed. And, but it is kind of like, it's weird in textbooks, like when there are like modern topics where a lot of the people are still alive, and they're kind of giving each other high five, and they want to, and so they kind of might do it in a way that's a little bit, you know, kind of, like, I agree, it's kind of like not really navigating it too smoothly through there. Yep, like that. Yeah, and then let's see, Hannah, what's one thing that you guys talked about back there?
We talked about the different checkpoints, so we have.
Yep. Yeah, and people like to call these checkpoints, and if you talk to somebody who's really into this, they'll say this. If you think about it, a checkpoint is actually when the cell looks internally to see how things are going. Like the G2 to M transition is a checkpoint where it's looking for DNA damage. The M phase spindle checkpoint is looking to see its chromosomes. And those people like to call start not a checkpoint, but really a control point where you're controlling the show externally, typically. You're saying go, like that. So a little bit of a detail, but yes, those three checkpoints. And you could see, like on this figure for intro bio, I say, you know, people love to call this a checkpoint. And you'll see there's a reason why we're going to, when we get in here and we're comfortable talking about it, we're going to drive in and say, it's a control point, but we want to talk about a little checkpoint that's part of it, where the cell's also looking internal, right? So the control point is PDGF going, push the button, start. That's what we really want to know. And when we get into chapter 20, maybe it's this chapter a little bit, but there's something we want to talk about where there's also a little controlling thing. The cell's looking internally to see how things are going, like that too. But yes, those three. Okay, and we gotta hang it up. So I'm gonna hand out your exams, and like I just said, dang it, I apologize that we have to take these exams, and they're hard.